how we can prepare juvies and how we can observe under microscope. So before doing experiment, I am going to give some general introduction that which method we are using today for making our vesicles. And we all human beings, animals, plants, and all, we have made up of cells, and a cell has outer boundary. We have discussed in all lectures that plasma membrane and cell membrane, it's very complex in nature because it contains component of cells. It contains proteins, carbohydrates, cholesterol, lipids, and all the, if we want to study the properties of this bilayer, so we have to reduce the complexity of this membrane if we want to study properties. So our membrane is basically made up of 50% of lipids, and other part is protein, and 5% of carbohydrates. So lipids is the basic main building block for any membrane. So now we are using phospholipid bilayer to make our vesicles. Let's go into the world of lipids. I will give the general introduction. This is phospholipids because it contains phosphate, and glycerol is basically the bridge between that binds the phosphate and fatty acids chain. It's amphiphilic in nature. Amphiphilic means that contains hydrophilic head and hydrophobic tail. Hydrophilic basically that loves the water, and hydrophobic that runs away from the water part. It's non-polar in nature, and it's likes the polar part of the water. So what happens when we put this into aqueous solution like water? So this part go towards the water, and this part it has phobia to the water, so it will arrange into the bilayer structure, closed bilayer, so that inside and outside is water. Head part is like this, is the exterior of the cell and interior, and this hydrophobic part is away from the water. So today we are going to make giant unilamellar vesicles. In this we can see like this is bilayer, black one is the bilayer, phospholipid bilayer, and inside is like this. Juvie is made up of 75 millimolar sucrose. So inside is sucrose and outside is sucrose solution. So we will form today juvies using lipid. We have to decide the lipid, and we have to decide the buffer for making the vesicles. So we will use L-alpha PC lipid today. PC is the head group and L-alpha is the fatty acid chains, and it will define the L is basically the L isomer of the lipid that define the stereochemistry of the lipid molecule. And in our lab we also this bilayer is this, and we also find the bending rigidity of the vesicle. Like in this video we can see this vesicle is fluctuating. So this fluctuation, these are thermal fluctuation, because inside is solute particles that is kicking out due to Brownian motion. They kicking out the wall bilayer, so they are fluctuating at room temperature. So we also find the bending rigidity for this bilayer in our lab, and all other properties like surface tension. Like what will happen if I change the outer solution, if I increase, like this is the vesicle, suppose it's making 300 millimolar sucrose solution. So inside and outside is sucrose solution. If I replace the outer solution with higher concentration, like inside is 300, if I put 400 millimolar sucrose solution outside, then there is osmolarity difference. And we know this is semi-permeable membrane that allow only the water molecule pass through it. Not sucrose and glucose, they can't pass through the membrane. So due to osmolarity effect, water will go higher concentration. So water will go from inside to outside, and the volume is reduced, and there is shape changes of the vesicle, which we observe all like pear shape, dumbbell shape, all possible shapes on morphology of the vesicle in our lab. So there are many methods to study the to prepare the vesicles like PVA, gel-assisted method, spontaneous swelling method, electroformation. We can prepare GUV from small unilamellar vesicles. We can form GUV from large unilamellar vesicles, and emulsion transfer method. So like electroformation method, in this we need a setup to give the electric field, like definite frequency and voltage we have to give for a like 4 hour, 5 hour. If we have like simple lipid uncharged like DOPC, PC, that is zwitterionic lipid, so we don't have to give higher frequency. But suppose we have complex lipid, like we have mixture of 2 or 3 lipids, like DOPC is zwitterionic, DOPS, like DOPS is negatively charged, or DOPG. So if we mix both these, then if we take, if suppose we are preparing only in sucrose solution, so that is also simple. If we want to prepare in ionic buffer, like there is potassium sulfate, magnesium sulfate, NaCl, so there is effect of ions also, and that charged lipid also. So this electroformation method does not work for the physiological conditions, buffer conditions. Either we have to give the higher frequency like 500 Hz, or we have to change the time, like normal in neutral lipid and simple buffer solution we have like 4 hours we can prepare. But if we have complex lipid and buffer also ionic, so we have to wait like 13 hours also. We have to keep higher frequency for the 14 hours. So it will be time taking process. So we can use alternative method, that PVA gel assisted method. This we can use for the complex lipids also. In this methods we have to take lipid. We have to firstly prepare the PVA solution, 5% PVA solution. PVA is a polymer, so we add 5 gram PVA in 95 ml water. We have to heat, for preparing 5%, we have to heat 90% so that it dissolve and make a transparent solution. So we prepare this. Then we have to take the glass slide and we have to coat the lipid bilayer, PVA bilayer, bilayer not, PVA layer, thin layer. So now we have to dry this PVA layer. So we kept the glass slide in the incubator for 30 minutes. Then after drying the PVA layer, we have to coat our lipid. So we will coat lipid. Now our lipid is basically, we store it at minus 20 degrees Celsius, because lipid is in powder form. We dissolve lipid into the chloroform for storage of the long time. So when we use that lipid to make vesicles, so we have to evaporate that organic solvent chloroform. So after coating the lipid, we will kept into the desiccator for the vacuum and evaporation for the organic solvent. Then after when the all evaporation happens, like then we will add our buffer solution on that PVA lipid coated PVA layer. Then after 30 minutes we will keep it, and then we will collect and we will observe our juice. So I will show the small video that how we can prepare, and then we will prepare our individual juice, and we will see that how juice looks like in the microscope.
So how long does it take to make the GUV in this particular process?
It takes half an hour.
Just half an hour.
Okay. So firstly we have to take glass slide, and this is our 5% PVA solution which we have already made. So we will take 250 microliter of that PVA solution, and then I am coating, like we have to coat this a thin layer of the PVA using the tip. Then after like 4 and 5 times we will homogeneously coating this. Then we have to keep it at incubator for 30 minutes at 60 degree. Then after 30 minutes we will take out, and then we will see like there is a thin coating of PVA on the glass slide. Then we have to coat the lipid. So For lipid, we use this Hamilton syringe. This is a special type of syringe. We can't use pipette because this chloroform doesn't work with the plastic. So this is the Hamilton syringe in which we have to wash this with chloroform first, and then we can take, and then after vacuum. This is our lipid, and then we will take our buffer. Today we are making in the water, so we will take water and then we will hydrate that lipid, so that because when we put this lipid into the solution, then due to hydrophobic effect, vesicles forms. So we will keep at 30 minutes, for then we will collect.
What is the concentration of the lipid?
1 mg per ml. We prepare our lipid, all lipids in the like
Volume? How much you inject?
10 microliter. So I will tell the amount all. Now we are preparing juice. So glass. So this is the procedure. We have to first preparation of PVA gel coated thin film on glass slide. Then we have to coat our lipid film, and then we have to hydrate that lipid film, and then we will observe our JV in the glass. So everyone is so everyone take one glass slide, and firstly you have to wear gloves.
दोनों हाथ पे चाहिए.
कहाँ वजन कोई नहीं करता, सबको दे दीजिए. लिक्विड ऐसे नहीं, मतलब जिस पोजीशन पे लिक्विड डालेंगे, ड्राई होने के बाद वी कैन सी द पैच, देन वी विल पुट आवर बफर ऑन दैट पोजीशन. So everyone take your glass slide, and you can write your name behind your glass slide so you can see your juice. So I will give a marker.
Just take your phone.
Cover slip.
No, the cover slip. Just take.
Pass yours so that I will give you.
So we keep it on something right? Because I think it might break.
Then
ऐसे टूट जाएगी. यूज सेलोटेप उसको थोड़ा सा लो और साइड से.
Measured.
Oh,
So
it's
it will
already
take
measured.
15. Okay. Then you can press and just.
दीदी एक और माइक्रो पाइपेट ले आओ. So you have to go like this.
सेल कॉम्प्लेक्स में किसी को भी पूछ एडी बनना
So this bubble like thing, na?
Push it to the.
Some kind of air or some.
move it to the edge maybe
remove on its own. So then I will keep it like this.
Then you are coating, so start from the other end.
Okay.
Okay?
I'll
Then
try to.
you can do easy.
यहाँ पे भी कुछ particles से दिख रहे हैं ना. It's like
From what?
ये करेगा इंटरव्यू.
No.
और इसमें ना मेरे को वो लेंथ जैसा कुछ जैसे नहीं पकड़े. Can you see it? Over here.
Actually, if it's in corner, it doesn't matter. We can coat the lipid into the like, homogeneous.
But these particle-like things that you can see, na,
Yeah.
doesn't matter. Chalo.
Is it okay?
Yeah.
Any match just to now these are to go into the incubator for half an hour, right?
So tomorrow they will have some other sort of experiments and हर रोज कोई experiments है.
You can put your glass slide vertically on that mark. We have to keep it on the opposite side of the
Oops.
Yeah,
Liquid
yeah.
is dripping.
We have to.
ऐसे डाल दूँ.
हाँ.
There you go.
हो जाएगा.
Did it break?
Oh,
हाँ, डाल.
I think the first few people will find it easy to put it in. बाकी लोगों को you know, they have
वो
to,
भी हाँ.
they can break other people's. I think I broke my.
But वो ऊपर से तो टाइट लग रहा है मुझे.
बन गए तो किसी ने.
ये क्या लगाया नहीं है ना. मैंने का इतना सफाई से किया.
लगाया नहीं अब तक.
I was very impressed too.
नहीं हमारा कुछ.
Theoretically, I can.
Somebody here needs the people.
Yeah.
Okay, now I'm nervous. Hey, don't press it completely. Think it is.
The important thing is to make sure your dominant hand is free for doing.
Don't know that. Just separate this out.
Otherwise, you're getting
What about
stuck
this
with
one?
this.
No, it has
It's a
elements
lot
of
tough.
because it will get
Only
into
at least
that.
your dominant hand is
Yeah, but this side doesn't work.
then it gets difficult to handle. You cannot shift in.
इसको भी कर देते हैं.
Why we are not putting directly the lipid on that glass substrate rather than everything?
They tell you this is the one you're going through. So okay, take care of it. Just know you can respect. Heavy light लगाओ, lens लगाओ, होता है.
From this react with lipid.
No.
And
हो गया?
जी एक सेकंड के लिए मुझे
रिकॉर्डिंग ऑफ करके पोजीशन देखना तो रखना होगा
It's almost as good as his accused like
We can observe under microscope. So before starting experiment, I will be giving the general introduction that how we can, which method we are using to prepare UV and all things like as we all know that human beings, animals, plants and all living organisms are made up of cells, and cells have outer boundary that is plasma membrane and all things, and that component of cell membrane, it's very complex in nature because it contains not only lipids, it contains proteins, carbohydrate, cholesterols and many other components except the lipid bilayer. So if we want to study the properties of this bilayer, so we have to reduce the complexity of the system like membrane. So like we know that our membrane is made up of 50% of lipids and other part is protein and 5% of carbohydrates and all things. So lipids are the very main important component of the membrane system. So we will be today we are making UV using this phospholipids. So we can this is the lipid. Phospholipid because it contains phosphate, and glycerol is the main bridge that binds that phosphate and fatty acids chain, and it's amphiphilic in nature. Amphiphilic basically that contains hydrophilic head and hydrophobic tail, and hydrophilic and hydrophobic basically these are the components. Hydrophilic that loves the water, and hydrophobic part that runs away from the water. It has phobia to the water. Now what will happen when I put this head and tail amphiphilic phospholipid into the water? So if we put in the water, so this head group basically it will go towards the water molecule, and hydrophobic part will run away from the water molecule. So when we put into the aqueous solution, it will form a bilayer. Like this is the exterior of the cell and this is the interior of the cell, and this is the hydrophobic part that is away from the water. This hydrophilic part upper and lower is in the contact of the water, so it will form like a vesicle. So in our lab now we are going to make this synthetic giant unilamellar vesicle. Unilamellar basically that contains only one bilayer. So in this picture we can see that like this is DOPC, this is the lipid or phospholipid. PC is the head group and DO is the tail group, hydrophobic part. So and this is made up in 75 millimolar sucrose. So for making UV we have to decide the lipid and we have to decide the buffer. Aqueous solution and lipid we have to take, and when we mix due to hydrophobic effect the vesicle formation will come. So this black one is the bilayer that is made up of like this type of structure, and in our lab we also study about the thermal fluctuations. Like this is the vesicle, and this is made up in sucrose, so there are solute particles. So due to Brownian motion, solute particle will kick the wall of this bilayer, and due to this there is thermal fluctuations happening. So we can see that this bilayer is fluctuating. So we can find the bending rigidity of this particular UV, that how much energy. Bending rigidity basically it's energy required to bend the bilayer. So how much energy cost required for bending this bilayer, we can find out this for every lipid like any buffer. So this is the vesicle. So now we have to prepare. There are many methods for the preparation of the vesicle. Like there is PVA method, spontaneous swelling method, electroformation. If we want to prepare UV, we can prepare small unilamellar vesicle, and then by swelling method we can prepare juvie from the SUVs, and we can prepare juvie from the large unilamellar vesicle to juvies also, and emulsion transfer method also. In electroformation method we have particular setup to give the electric field, like we have to give frequency and voltage, particular frequency voltage for like 4 hours 5 hours to prepare the juvie. But like there are a number of juvies, like charged, uncharged juvie, like DOPC. This is deuterionic juvie, like neutral, that has no charge. When we prepare juvie, it depends only upon two factors. What is our buffer? What is our lipid? If our lipid is neutral, like DOPC, that's neutral lipid. And if we prepare our DOPC juvie in the sucrose solution, so it is like simple method, like there is no charges, so we can prepare juvies easily. But like if I am taking charged lipid, like DOPC is neutral, I am adding DOPG. DOPG is negatively charged lipid. So I am mixing 2 or 3 types of complex lipids, and I am trying to form the juvie. So electroformation method, it will take, like we have to give the higher frequency, 500 hertz or more, and it will take more time, like we have to keep it for 13 hours overnight, 12 hours, so it is taking large time to prepare juvie in the complex system. So we can use alternative method, PVA gel assisted method. In this basically we have to prepare 5% PVA, and we have to coat thin PVA layer on the glass slide, then we have to dry it, then we can coat our lipid, and then after evaporation of organic solvent we can put our buffer, and then it will take like half an hour or 2 hours maximum, so we can prepare our juvie using this method. So there is small video.
ये जो like when you say buffer, what exactly?
Buffer, it can like sucrose solution, if we want to prepare in
which you are putting, is it?
yeah,
Okay.
our lipid, and buffer is our aqueous solution in which we are putting our lipid so that it can form vesicle. And like in previous picture, like inside is sucrose solution, outside is sucrose, that's water completely in fluid system. And we can see that only black bilayer, complete closed sphere. So inside is a buffer solution, outside is also a buffer solution. So this is the video, small video. So for preparing juvie today we are going to prepare using this PVA gel assisted method. So we will take glass slide, and then we have to take this 5% PVA, and we have to take 250 microliter of PVA, and we will coat a thin layer of PVA on the glass slide, and then we will keep that incubator for 30 minutes so that it can dry, and then we will coat our lipid. Like we can put in the incubator, then we will take it out, and we can see like there is a thin film of PVA is on the glass slide. So now we have to coat the lipid on this dry PVA layer. So we can coat our lipid and in the drop form, and then we will kept it in the desiccator, because this lipid solution we kept at -20 degree. We dissolve this powder form of lipid in the chloroform for storage. So when we are preparing juvie, we have to evaporate this organic solvent. For this we have used vacuum, and then after the lipid, after the vacuum, then we will hydrate using our buffer solution. Then we will keep for 30 minutes, and we will collect this, and then we can observe our solution juvie in the microscope. So everyone have glass. Using that.
How do you know it's 250?
No setup.
Acha.
So you have to make this homogeneous thin. PV 5%. So you can take Divanta. एक वो छोटे वाले लोग को आ जाएंगे, तो इनको उधर ही लेके इनके क्लास में उधर ही रखवाते हैं। These are the many types of lipids like unilamellar and multilamellar. Unilamellar basically small, large and juice, and in this multilamellar and oligolamellar. So in multilamellar we can see this type of there are many bilayers formed. So these are multilamellar, this is unilamellar, and these are small and large unilamellar vesicles, because we have to sonicate the large unilamellar vesicle in the sonicator to form small unilamellar vesicle. And for small unilamellar vesicle we use this rotary evaporator. Basically we have to put our lipid and then we have to rotate, and for getting the thin film, and then if we hydrate this, then we can get this small unilamellar vesicle. And then from small unilamellar vesicle we can prepare juvies, also giant unilamellar vesicle, using the electroformation, but for the long time, because we need swelling of this small unilamellar vesicle. This method from small unilamellar vesicle to or large unilamellar vesicle we prefer only if we want protein in our juvie. So we can't put protein directly in the juvie. So we form small liposomes like small unilamellar vesicle or large unilamellar vesicles. We put protein in that, then we can form from this proteoliposome to juvies. So it will directly contain that protein in the bilayer. So this is used for the transportation of anything, if we want. Because protein, the work with protein in the juvie is very difficult to put the protein. And it's we can't we have to store protein at physiological conditions, 4 degree. We can't put at room temperature for even smaller more time. So we have to take care of all things, so that if we prepare these small liposomes using protein, we can store it at -80 for longer time, for 6 months. So we can store, but juvie we can't store. We have if we prepare juvie, we have to use 2 days or in 3 days. After that they are degrade. Okay. So that's why for proteoliposome we can use basically this method, and we store at -80, and then we can.
You want the juvie,
Yes.
basically use
Yes.
the electroformation.
So for electroformation is the same. We have to take out our lipid from -20 degree, and we will wait up to at it gains room temperature. Then same we have to coat that lipid on the glass slide. Same here our ITO coated plate is substrate, and there PVA coated this substrate. So we have to coat our lipid, and then we have to use vacuum for solvent evaporation. Then we will make chamber, and we will fill our buffer solution, and then we will apply sinusoidal. This is our protocol that we have to change after 10 minutes. Because first step is we give low frequency. It's only for hydration, and the next step we will increase voltage for the swelling. and then at the end we will reduce frequency so that the swell vesicle detach from the glass slide. So basically in electroformation, three main steps is play important role, that hydration, swelling, detachment. Then this is also like video. First step is coating lipid. So we have to wash first this Hamilton syringe with chloroform at least 10 times, so that like it is super clean. It should be super clean. So we will wash and we will take our lipid. Now we will take lipid. These are the ITO coated glass plate. This is the copper strips. So we can put our like electrical signal, complete the circuit. So now we can coat our lipid. This we can see on the both glass slide we can coat.
कितना volume?
10 microliter. So then this is the vacuum step. So we have to leave it for 30 minutes so that all chloroform evaporates completely. Then after vacuum we have to make chamber. Now we have to hydrate this dry lipid film. So we will make chamber. We will take this glass slide, and this is Teflon spacer. Basically we will put on the coated part, and then we will put this so that this lipid film face each other. We will make chamber and we will close it by using these clips. And now there is a hole in the spacer, so we can fill the buffer solution in this chamber. Like, so we will take our buffer solution using syringe. Now through this hole there is a tiny hole, so we can fill, like we can see buffer solution is big. So now this is the like solution filling part, chamber preparation. So we have to, now this is chamber, hydration is complete. Now we have to apply our AC signal. So we will attach the positive and negative part of this through this copper, and we will kept in the incubator. We use incubator basically because every lipid has their transition temperature. So we have to, because lipid at low temperature, lipid is in gel phase. So for making membrane, we need fluid phase. So we have to keep in mind the temperature, melting, that phase transition temperature for every lipid. So we have to put the higher temperature so that it remains in the fluid phase. So this is our setup. So from this we can give frequency and we can give this voltage, and then we can apply for like 4 hours. We are changing like after 10 minutes, 20 minutes, changing the frequency and voltage. If there is like complex system, so we get this frequency, like this is for neutral, this is 10 Hz. For charged lipid or physiological buffer, so we get this 500 Hz. And for overnight, like we will leave it overnight, and then in the morning we will collect and then we can observe. So this is the electroformation method. Now time is happening, so we will coat lipid on that our glass slide. L-alpha PC. So it is now at room temperature.
Okay, so you have taken it normal.
From -20 before 30 minutes.
Okay.
So
If there is some leftover, can you actually put it back, or is it okay to cycle it up and down in temperature?
yeah, we can use like after coating we immediately put this parafilm, and then we can put
Put it back in
-20.
the minus.
Yeah. Then we can use like 1 year also.
Alright.
We make Stokes solution, 1 mg per ml Stokes solution, and we are using it 1 year.
1 year.
Then after 1 year it can degrade. slowly. So we have to finish it.
So you
maximum.
kind of make a mark and
Yeah, we write date basically on what date we are preparing our stock solution. Then we are using many times, like if it is older, then we are making, then juice is not forming. So we
And you have to
confirm that
just-
it's
gone. So
Yeah.
you have to discard it.
Yes. So in glass, your name.
ऐसे उठा लो
ऐसे हाँ
टूट गया
नहीं
अच्छा ठीक है
you can take your
इतने आ जाएंगे अंदर सब
ग्लास स्लाइड ले लो फिर लिपिड कोट करवाने
पहले बाकी उठा लो मुझे पता नहीं मेरी कौन सी है
So just take it and find your name and just put it.
so that
Yeah.
we have to
बाकी
take
तो भी दे दो.
क्या करना है
time
Glass slide होगी.
so we can see from this mark
नहीं मैं तो ले चुकी हूँ मैं तो उधर देख रही हूँ
so like this upper is 10 so now we have to coat so like there is homogeneous coating so we don't need to coat
Coat
this
that
side.
side. Yeah.
Hmm.
Zoom out, yeah.
So zoom out firstly.
So you just want to put tiny drops.
And then you can, I can coat it this way. Now we can
Alright. That's okay.
ये प्लीज पहले रख दीजिए सॉरी
Should I put it back into the? Because I think you
Yes.
me.
इससे डबल हुआ है ना ये से डबल
Yes.
ऐसे देख के भी पता चलता है ये नाम से ही पता चलता है
Naam se we have
वैसे
to
नहीं
mark.
देख के नहीं पता चलता
Yes, we can like this is
Yeah, because there's some
Yeah.
gathering.
You can also look at the
Yeah.
reflection of the name we put there.
Yeah, the name of course. So but she was asking whether without the name can you figure out. So she
Yes.
said you can see that the
There's
excess
a rank.
excess has formed that ridge. So then you know which is the side that's good. Nice. Yeah, you can micro and then these people come in. आप लोगों को अंदर जाना है तो you can just go ahead. Everybody has a slide.
This is usually liquid, right?
this is array copies.
जो
So we have to post.
It's not drawing.
Yes.
बताओ क्या पढ़ना है
तो थ्योरी में याद तो होना चाहिए ना क्या बताओ पहला स्टेप क्या है अभी वैक्यूम करना है ना हमें क्योंकि इसमें क्लोरोफॉर्म है आधा घंटे में बस दो ही
Second method that she was telling us about of making electroformation. Thank you. I couldn't get an electroformation. You will look. Thanks. Sorry to interrupt. So frustrating.
मतलब ना बहुत
The point of the accumulation because there's no this thing, no open
प्योर नेगेटिव
नहीं मतलब मिक्सर बनाना है
20% नेगेटिव ना बताओ मैक्सिमम नेगेटिव चांस उसके ऊपर
They basically push air out. The whole table is actually floating. So what happens is that if you touch one thing, so then there's a pressure adjustment that happens. Either pumping happens or something is let out so that the vibration is not just transferred anywhere. So, yeah, there's an air compressor, you can see that. And these legs are like, so first of all, I think these tables are completely heavy so that no vibration can be felt.
So you use Hamilton instruments for only vesicle making or some other stuff also?
for like chloroform, ethanol, these things.
Any liquid which can react with
Plastic.
I'm asking only for vesicle systems you are using or any other system that you are going to do here in this lab?
Actually, we are only
If you are using manual systems, you are not allowed to touch the tip using
We have from sample form. From the basics.
What we have is from Colmar.
Alma
But I think solar is more expensive. That was the cheaper option for us.
But Aldous does not provide a customizable solution. They have DIY methods.
And how do you make sure that vacuum is sufficient in creating it is not lock, that's okay. But the pressure that is building there is sufficient.
Actually, we tried this for 30 minutes now, and then
You try water boiling using this.
saturation pressure, like for complex buffer, we keep NaCl solution for the saturated
So
buffer
it
pressure.
boils.
Yeah, micrometer. So 1 micrometer is also visible.
जो जब फ्लक्चुएशन वगैरह स्टडी करते हैं उसके लिए जायंट
जायंट ही चाहिए क्योंकि ये इतना छोटे का तो हम बस ये देख सकते कि हाँ हमारे पास कुछ है लिपिड मोलिक्यूल्स है बट वी कांट स्टडी द फ्लक्चुएशंस पार्ट सो वी हैव टू प्रिपेयर दिस लार्ज वन इन द माइक्रोमीटर रेंज सो दैट वी कैन स्टडी द दिस
स्वेलिंग के लिए कौन सा मेथड
Yes, electroformation method. So in this method, we give basically electric field and voltage. Frequency we have to give. So firstly we have to take out our lipid stock solution from -20°C. Then we will wait until it comes at room temperature. Then we will coat this lipid film on the ITO coated plates or platinum wire chamber. There are many chambers for the electroformation. We can't only like coat on the glass slide like we did in PVA method. So we will take that ITO coated glass plate, we will coat our lipid, then we will keep in desiccator for evaporation of solvent, and then we will fill our solution, buffer solution, and then we will apply our sinusoidal film. Like these are the basically steps. First step is the coating lipid. So for this we have to wash our Hamilton syringe with chloroform so that it should be super clean. So then we will take our lipid. After washing, then we will take lipid solution, and these are the ITO coated plates. This is copper strips at the corner so that we can connect our positive and negative, and we can complete the circuit and we can apply the.
So the vesicle is charged or like how is it swelling like the swelling effect how is it?
Okay, this is like first we have to coat. Then like I am coating now. You can see this lipid coating. Then coating part is done, the solvent evaporation part. So we will keep our glass slide in the desiccator, and then we will left it for 30 minutes, and then we will make the chamber for hydration, because on the glass slide we can't directly put our buffer solution. So for chamber formation, like this is glass slide and this is the Teflon spacer, white one. We will put on the coated part, then this another glass slide, and then we will make chamber, like coated part will face each other, and then we will close using these clips. So this is the chamber formation. Chamber is closed. Now there is a hole in this Teflon spacer. So using this syringe, we will fill our buffer solution, and then we will fill this chamber through this hole in the Teflon spacer. So there is a hole, tiny hole. So using syringe, we can fill, like we can see the filling of buffer solution inside this chamber. Then complete filling. It will take like 2 ml of solution. So now we have to apply AC circuit. So we will connect with the positive and negative part with this copper strip, and then this will complete the circuit, and then we will apply the frequency and voltage using the setup. This this is the set. Like from this we can give frequency, and this is the voltage, then we can connect and we can on this, and then we will keep for 4 hours. So then using syringe we will take it out after 4 hours through the same hole.
After, like, it's after you connect the circuit, it's already swelled. स्वेलिंग ये वाला पार्ट में हो रहा है ना लास्ट में.
हाँ, अभी फर्स्ट पार्ट में हाइड्रेशन होगा. जब हम 10 हर्ट्ज हमने फ्रीक्वेंसी दे दिया, वोल्टेज दे दिया, तो 30 मिनट्स तक हाइड्रेशन होगा पूरा कंप्लीट. फिर उसके बाद स्वेलिंग स्टार्ट होगी. स्वेलिंग के बाद लास्ट वाले पार्ट में डिटैचमेंट होता है कि जो भी वेसिकल बन गए हैं, जो हमारा ग्लास स्लाइड से स्टिक है, तो फ्रीक्वेंसी मान लो 10 हर्ट्ज है, लास्ट वाले पॉइंट पे मैंने 4 हर्ट्ज कर दिया, तो जो वेसिकल है वो डिटैच हो जाएंगे ग्लास स्लाइड से और बफर सोल्यूशन में आ जाएंगे. तो लास्ट पार्ट इज यानी हाइड्रेशन, स्वेलिंग एंड डिटैचमेंट.
जो ये दोनों है चैम्बर प्रिपरेशन और अप्लाई एसी सिग्नल, वो आप ही होगा या
नहीं, क्योंकि इसमें टाइम लगता है, तो इसलिए वी यूज PVHM assisted method, because it takes 4 hours, so we have only 3 hours for lab session. So that's why I am showing the video that how you can prepare this.
और जो वो एसी सिग्नल देने के लिए जो मशीन यूज होती है वो कौन सी होती है?
वो अंदर रखेगा. There are big balloons like this. Okay.
Multi-laminar.
Yeah, and like in this inner box also like
There is one
multi-recipe.
Yeah,
This
oh,
one
okay.
area, like in
And
this
here also we can see,
yeah. So we can see all types.
All types.
All sizes, all types. So now in this like inside water, outside water.
They are not moving, right? Why should they be moving? They are moving.
And you can see fluctuations also there is happening at once.
Oh yeah. Okay.
So you have to focus
No, it's moving. It's moving.
this one, and for this we can focus on one time at one vesicle. Like this one, of our input. You can move your vesicle.
This unilaminar one or
we can take snap and we will now for the study, like this is the simple system. Outside is water, so we can put any other copper solution from outside, and then it will replace the outer solution. Like now outer is only water. If I put glucose, so there will be outside glucose and
And the
inside
shape will
water.
change.
So water will go outside,
and it
so
will change.
we can see the shape changes.
Okay.
So we can record and we can make live videos of the shape
Shape
changes,
changes.
and we can study the morphology.
Thanks. Nice.